Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
Defying time. The science and innovation behind longevity and anti-aging. The quest for longevity and the secret to delaying the effects of aging have fascinated humanity for centuries. From ancient alchemical experiments to modern biotechnological advancements, the pursuit of a longer and healthier life has always been a driving force behind scientific curiosity and innovation. In recent decades, the field of longevity and anti-aging research has undergone a profound transformation, fueled by breakthroughs in molecular biology, genetics, and regenerative medicine. Researchers are no longer just studying the symptoms of aging. They are diving deep into its root causes, unraveling the intricate biological processes that govern the passage of time in the human body. At its core, aging is a complex and multifaceted process, shaped by a combination of genetic, environmental, and lifestyle factors. Scientists have identified a range of cellular and molecular mechanisms that contribute to the aging process, often referred to as the hallmarks of aging. These include genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, and mitochondrial dysfunction, among others. Each of these hallmarks represents a layer of complexity in how cells and tissues deteriorate over time. For example, telomeres, the protective caps at the ends of chromosomes, shorten with each cell division, eventually triggering cellular senescence, a state where cells stop dividing and start releasing inflammatory signals. Understanding these mechanisms has been pivotal in identifying potential targets for intervention. One of the earliest and most promising areas of longevity research is the study of caloric restriction. Observations dating back to the early 20th century revealed that reducing caloric intake without causing malnutrition could extend the lifespan of animals such as mice, rats, and even primates. These findings led to the hypothesis that caloric restriction might activate protective pathways in cells, enhancing their ability to repair damage and maintain homeostasis. Studies in this area have uncovered key molecular players, such as the sirtuin family of proteins and AMP-activated protein kinase, AMPK, which regulate energy metabolism and stress responses. Compounds like resveratrol, found in red wine, have been studied for their ability to mimic the effects of caloric restriction, sparking interest in developing pharmacological interventions for longevity. Another groundbreaking development in the field has been the discovery of senolytics, a class of drugs designed to selectively eliminate senescent cells. Senescent cells, often referred to as zombie cells, accumulate in tissues as we age and contribute to inflammation and tissue dysfunction. Research has shown that clearing these cells in animal models can improve healthspan and delay the onset of age-related diseases. Senolytic therapies hold immense promise, not just for extending lifespan but also for alleviating conditions such as arthritis, cardiovascular disease, and neurodegenerative disorders. The study of genetics and epigenetics has further revolutionized our understanding of aging. The identification of longevity-associated genes, such as FOXO3 and SIRT6, has provided insights into how certain genetic variations can influence lifespan. Meanwhile, epigenetic changes, such as DNA methylation and histone modification, are being studied as both markers and regulators of aging. Epigenetic clocks, which measure biological age based on patterns of DNA methylation, have become valuable tools for assessing the effectiveness of anti-aging interventions. These clocks are helping researchers test whether treatments like caloric restriction, senolytics, or gene therapies can truly reverse or slow down the aging process. Stem cell research has also emerged as a cornerstone of longevity science, offering the potential to regenerate damaged tissues and organs. Advances in induced pluripotent stem cells, iPSCs, have opened the door to personalized regenerative medicine, where a patient's own cells can be reprogrammed and used to repair age-related damage. Moreover, the development of organoids, miniature, lab-grown versions of organs, is providing researchers with unprecedented opportunities to study aging and test interventions in a controlled environment. As the understanding of aging deepens, so too does the realization that it is not a uniform process. Different tissues and organs age at different rates, influenced by factors such as metabolism, immune function, and environmental exposure. This has led to the concept of precision aging, where interventions are tailored to address the specific needs of an individual's body. Such an approach is particularly relevant in tackling age-related diseases, which often emerge due to the interplay of multiple biological systems. For instance, cardiovascular aging involves not just the stiffening of blood vessels but also the accumulation of lipids and the decline in mitochondrial function, requiring a multifaceted strategy for prevention and treatment. Longevity research has also sparked interest in understanding the role of the microbiome, the trillions of microorganisms living in and on the human body. Studies have shown that the composition of the gut microbiome changes with age, influencing metabolism, immunity, and even brain health. Researchers are exploring how modulating the microbiome through diet, probiotics, or fecal microbiota transplantation might promote healthy aging and mitigate the risk of diseases such as Alzheimer's and Parkinson's. The journey toward understanding and extending human lifespan is as much about quality of life as it is about longevity. Aging is often accompanied by a decline in physical and cognitive function, making the concept of health span, the period of life spent in good health, equally important. Researchers are increasingly focusing on interventions that enhance health span, aiming to compress the period of morbidity and ensure that people not only live longer but also live better. This shift in focus underscores the ethical and societal implications of longevity research, as extending life without improving its quality could pose significant challenges. The second wave of advancements in longevity and anti-aging studies is being driven by breakthroughs in biotechnology, artificial intelligence, and personalized medicine. These innovations are pushing the boundaries of what was once thought possible, with scientists aiming not only to delay aging but also to reverse its effects. Among the most ambitious approaches is gene editing, particularly through the revolutionary CRISPR-Cas9 technology, which enables precise modifications to DNA. This tool has opened up possibilities for correcting genetic mutations associated with age-related diseases and even enhancing cellular resilience to the aging process. Researchers are exploring ways to target genes involved in DNA repair, oxidative stress resistance, and cellular regeneration, laying the groundwork for potentially transformative therapies. One of the most intriguing areas of focus is the concept of cellular reprogramming, inspired by the work of Nobel laureate Shinya Yamanaka. By introducing a set of specific factors, now known as Yamanaka factors, scientists can revert adult cells to a pluripotent stem cell state, effectively resetting their biological age. Recent studies have shown that partial reprogramming, which avoids complete dedifferentiation, can rejuvenate cells without compromising their function. This approach has demonstrated promising results in animal models, where aged tissues have regained youthful characteristics and resilience. The potential of reprogramming to combat aging and regenerate damaged organs is immense, though its safety and efficacy in humans remain areas of active investigation. In parallel, Advancements in bioinformatics and artificial intelligence, AI, are accelerating the pace of discovery in longevity research. AI-powered algorithms are being used to analyze vast datasets, uncovering new biomarkers of aging and identifying novel drug candidates. These technologies allow researchers to simulate and predict the effects of potential therapies, reducing the time and cost associated with clinical trials. AI is also being utilized to optimize personalized treatment plans, ensuring that interventions are tailored to an individual's unique genetic, epigenetic, and metabolic profile. One notable development in the pharmaceutical realm is the emergence of rapalogs, a class of drugs derived from rapamycin. Rapamycin, originally discovered as an antifungal compound, has been shown to extend lifespan in multiple species by inhibiting the mTOR pathway, a key regulator of cellular growth and metabolism. While its use in humans is still under investigation, early studies suggest that low-dose rapamycin or its derivatives could have anti-aging effects without significant side effects. These findings have sparked widespread interest in developing next-generation mTOR inhibitors as potential longevity therapies. The immune system, which plays a central role in maintaining health and combating disease, has also become a critical target in anti-aging research. With age, the immune system undergoes a decline in function, a phenomenon known as immunosenescence. This decline contributes to increased susceptibility to infections, chronic inflammation, and cancer. Researchers are exploring ways to rejuvenate the immune system, such as using thymic regeneration therapies, which aim to restore the function of the thymus, a gland critical for the development of T cells. Other strategies include developing vaccines tailored for older populations and using immune-modulating drugs to reduce age-related inflammation, often referred to as inflammaging. The intersection of aging and brain health is another focal point, given the profound impact of neurodegenerative diseases like Alzheimer's and Parkinson's on aging populations. Advances in neuroscience have shed light on the mechanisms of cognitive decline, highlighting the role of protein misfolding, synaptic dysfunction, and neuroinflammation. Researchers are investigating therapies that target these pathways, including monoclonal antibodies to clear amyloid plaques, small molecules to enhance synaptic plasticity, and lifestyle interventions such as cognitive training and dietary modifications. Moreover, the potential of brain-computer interfaces and neuroprosthetics to mitigate the effects of aging on the brain is an exciting frontier, offering hope for preserving cognitive function well into old age. Diet and nutrition remain foundational elements of longevity science, with ongoing studies exploring the impact of various dietary patterns on aging. The ketogenic diet, intermittent fasting, and plant-based diets have all been associated with improved metabolic health and extended lifespan in animal models. Researchers are working to translate these findings into practical dietary recommendations for humans, with a focus on optimizing caloric intake, macronutrient balance, and micronutrient density. Nutritional supplements, such as NAD+ precursors and antioxidants, are also being tested for their potential to enhance cellular energy production and reduce oxidative damage, though their long-term efficacy remains under scrutiny. Social and environmental factors are increasingly recognized as critical determinants of longevity. Studies of blue zones, regions where people live significantly longer than average, such as Okinawa in Japan and Sardinia in Italy, have identified common lifestyle traits, including strong social connections, regular physical activity, and diets rich in whole foods. These findings highlight the importance of a holistic approach to aging, one that addresses not only biological but also psychological and social well-being. As the field of longevity advances, ethical considerations have come to the forefront. Questions about access, equity, and the societal implications of extended lifespans are driving important conversations. Who will benefit from these therapies, and how will they be distributed? What are the economic and environmental impacts of a population living significantly longer? These issues underscore the need for a balanced approach to longevity research, one that considers not just the science but also its broader implications. The pursuit of longevity is no longer confined to the realm of science fiction. It is an active and rapidly progressing field with the potential to redefine human health and aging. From cellular reprogramming to AI-driven drug discovery, the tools and technologies being developed today could pave the way for a future where aging is not an inevitable decline but a manageable and reversible process. However, the journey is far from over, and the challenges ahead are as complex as the biological systems researchers seek to understand.